Okay, so actually, I want to start off first with a couple of PowerPoint slides just to give you sort of to reiterate what Les just said. Um, so start there. I'm going to, in that, shoot, sorry, I have to get it over on the right screen. No, actually, I won't show it in presentation, but well, I better. So this is just a little bit of introduction into how you use virtual cell. And again, as Michael pointed out early on, I just want to remind everybody that we are funded by NIH, by our 24 program, along with COPASI. And as Les said, this is the problem domain for virtual cell. So the problem domain that we solve is reaction and diffusion, right? So the divergence of the fluxes and the reaction rates. So within that problem domain, though, we can have a multiple different ways of solving the equations depending on the system, as Les just explained, whether it's stochastic versus deterministic, for example. So these basically show you the different types of problems that virtual cell can solve. So as Les said, you can start with a simple ordinary differential equation if you're not going to include diffusion. And along with, in the case of ODEs, we can also do some estimation of parameters. For example, if you don't know a couple of rate constants and you want to determine what they might be based on some experimental data. So we can estimate parameters, which we do actually using COPASI's tools. So you'll see more of that from Pedro Mendez in talking about COPASI. We can not do it deterministically, but determine it using stochastics, again, in non-spatial situations where we're not worrying about diffusion. And in addition, I'm going to go back around the other way. First of all, we can do it including space on the other side. So we can use partial differential equations to include diffusion and the space involved, which I'll show you as well. We can also do it stochastically in space using a solver package that we've modified a bit called Smolden, which accounts for particles in space. And then we can also do, as Les said, hybrid. That is, we can have some molecules be deterministically solved and some molecules be solved by probabilities using spatial stochastics. So I'll show you a brief bit about that as well. And also I wanted to point out that we can start by building the model with two different strategies. One is a classic sort of network diagram where we define the molecules and the kinetics for the reactions. And I will present that this morning. And then we can also do this in a method called rule-based modeling, which defines the reactions that will occur based on rules. And Michael Blinoff will talk about that a little bit later, I think tomorrow. Yes, tomorrow. We'll talk about how to use the rule-based approaches, right? And the rule-based approaches also allow for another method of solving the problem, which is called network-free simulations, in which you don't build a biological network yet. You just create the molecules as you need them, or the species as you need them, basically. And again, Michael will explain more of doing simulations using this network-free approach as well.
So it will be today, 3:15.
Oh, is that today? Oh, yes, I'm sorry, that is today. This afternoon. My schedule is partially blocked on my screen. So yeah, this afternoon, Michael will talk about that. And also, I think Wednesday, Jim Schaff will talk about the fact that we can also do some kinematics. That is, most of what I've talked about so far, the geometries cannot change. So once you create the geometry, the space, that's what it is. And things don't move, just the molecules convert back and forth or move by diffusion, but the geometry itself doesn't move. We do have the ability to solve problems in 2 dimensions where the geometry itself can move, and that's called kinematics. And so we can do partial differential equations plus kinematics. And Jim, that's what's shown in the little movies, that we can move the cells around. And Jim Schaff will talk about that briefly, I think, on Wednesday. Okay, so in order to really understand how to build models within virtual cell, you have to understand a little bit of the philosophy behind which it's built. And so it was built largely by cell biologists from a cell biologist's point of view, that is. So the first thing that we do is we build a mechanism, the physiology. What are the species involved? What are the reactions? How does one molecule convert to another molecule? And where are they located? What compartments are they in? And so we'll build that part of the model first, and we call that the physiology. So before we do any sense of how we're going to build the mathematical model, we're going to build what we think the biology is. And so once we start with the biology, then from there, from that one biology, you can apply many different methods for solving the problem. You can do it deterministically or stochastically, and have multiple different ways of solving it in those two situations. So we can use ordinary differential equations, or we can put it in a different space, or we can put it in multiple different cell types. So we build one physiology, and then from that we make multiple applications of the same. And I'm going to show you that in detail in the next hour. So from one physiology, we get multiple applications of that model that are all stored together in a database. And each application then will be able to be uniquely described in the math. So virtual cell will automatically take the application and build the math description for it. And you can see the math description, you can save it out, you can even build your own, you can edit it and make a new math description if you want, but there's one math description for each application. Okay? If you do something that's going to change the math description, that becomes a new application. Okay. So there's a one-to-one correspondence between the application and the math description. And then from there, from each math description, you can make multiple different simulations. You can change how long it runs or how fast it runs, or the mesh size if it's a PDE. So each application will have multiple different simulations. And you can also change parameter values and things like that, and variable values. So that's kind of the basis of a biomodel in the virtual cell, and we store them all together within a central database here at UConn. And so if you use our solvers, I'll show you this in a few minutes, the model will be stored in total with all these component parts and any results that you have in your simulations on the servers here at Virtual Cell, and you can access it. So just a brief overview of the graphical user interface first before I just launch into building a model. And also, I do want to point out, as Les said, this is going to be very similar to what's online. So please feel free to break in and ask questions at any time. It always is more interesting when it's a discussion. But just a brief overview of the sort of workspace that you're going to be dealing with in Virtual Cell. So we have divided up the workspace into 4 different panes. So there's one window, and it has 4 different panes. The upper left side of the pane is the navigation of your own model. Whatever model you're working on right now is navigated in this window in the upper left side. If you go to the upper right side, this is the main workspace, which shows, if you're working in the physiology, the reaction diagram or the list of simulations. Sort of the main space of where you're going to do a lot of the work. The lower right-hand side is properties of whatever is selected, whether it be a model or a component in the model you're working on. The individual properties will be down in this lower right-hand side. And so this will be where you can, for example, add specifics about the kinetics of a particular reaction that you're looking at. So this is a properties window. And there are other tabs in this window. For example, it will show any errors or warnings that VCell has, which can be very useful when you're building the model. And it also has a link to annotations, and I'll try to show that as well. Okay. And then finally, on the lower left, as I said, if you save the model to our database, you can save it as a standalone VCML model, but it's most convenient to save it within the database. Then there's a list of all the models that are associated with the database on the left side. So these are all the resources where you find models. So it will list your models, models that are private to you. It will list models that somebody else has shared with you. And so there's a way of sharing models with individual people. We have built a bunch of tutorial models, and we'll go through one of those today, which are to help you learn how to use various aspects of virtual cell. And then you can make models open to the general public. And we have a couple categories of those. Primarily, if you publish anything using a virtual cell model, please let us know, and please make your model public so we can publish it. And that means that anybody who reads your paper can open the model and play with it, save it to their own, change it, do whatever they want with it. They'll have full access to the model. They can't change it. It becomes permanently frozen in time to match your publication, but they can save it as their own model then and go on and work with it. And then we also have some models that are not published, but we've curated. Usually they've been published in another form, maybe a Copazi model or some other form of mathematical model that we've translated into a virtual cell model. So there is a peer-reviewed publication about the model, and then there's uncurated models, which is just anybody, often they're students in a class. So, you know, they're whether they're good models or not is up to you. We make no claims as to how good they are. Okay, so that's my little introduction into the user interface. So I'm just going to go right now to the interface. Again, here are my 4 window panes. And the model I'm going to describe to you and showing you how to build models in virtual cell, I'm going to start with just the physiology of the model already made so that you know where we're going, and then we'll start on a blank page. So the model I've chosen is one of the tutorial models. So if I go to the model database and I look down here, this is the tutorial multi-app. And as you can see, I've clicked on it, so I get all the information about this. And if there were any annotations, you would see them, but we haven't annotated the tutorial models yet. That's interesting. So should go back there. Okay. So anyway, this tells you all about the provenance of the model and how many applications there are and what kind of applications there are. So if you're looking for particular types of models, you can get that information here. So I've opened up this model. The tutorial models also all have a PDF tutorial that you can follow along online. So if you look at our website, vcell.org, you'll find all of the tutorials there. So they're built to basically help teach certain components of VCell. So what we're going to build today is a very simplistic model, biomodel, that is really designed to show you the overview of all the the types of features and types of models that virtual cell can do. And it's a simple transport cytoplasm to the nucleus for molecules that require the transporter RAN. So it has 2 species that are components of the model, RAN, which is a chaperone that helps bring molecules through the nuclear envelope, and a cargo molecule. We don't care which one, so we just call it cargo, so it's C. And when RAN binds to C, it forms a complex of RAN C, RAN with cargo, and that complex is able to flux through the nuclear pore. So it will go into the nucleus, and then therefore you'll get the complex of RAN-C in the nucleus. So that's the model we're going to build. Again, it's a little bit of a toy model just to put all the individual parts in it so you can get a good overview of VCell. It's not terribly biological at the moment because nuclear transport is a much more complex problem. But that's where we're going to go, just so you know what we're trying to end up with at the end. Okay, and then we've built a whole bunch of applications, and I'll show you how to do that are different applications of this particular biology. So let's start with trying to build the biology. And again, if there's any questions, just feel free to jump in. Or if I'm going too slow, feel free to jump in too. Okay, so I'm going to build a new model. Okay, so I'm going to get a new biomodel going. And it's going to go on my other screen, so I have to pull it over. That one. Okay, so when you create a new biomodel, you end up with a blank screen. Across the top are the major tool menus. So for a file, you can create a new one, you can open an old one, you can save it as. It's important to see save as local. So if you want to save this model not to the database, but to our own computer, your own computer, you can save it as a VCML file locally. That will become important later on. And then there's a couple of other menus to import and export the models that I'll talk about a little bit later. You all have an account, I hope, in the VCell, and in order to have access to the database. And so this menu is just to deal with the users. You can change the users, or sometimes you'll need to reconnect if there's been an interruption in the internet service. The window we don't have to worry about. There are a couple of tools that are here, which again are a little more esoteric and we can talk about later. And then most importantly is the help menu. We have a very extensive help menu for VCell. So if you go to help and VCell help, you will end up with a help menu. Sorry, I'm using a different screen than my basic screen. That provides extensive information about everything. So if you want to find something about simulations, let's say, you can go here and you'll find descriptions of everything to do with simulations. So there's lots of stuff that you can see. So it's fully searchable and it's very extensive. So if you have any questions, I strongly urge you to look in the help. It actually does provide help. And then there's also a little bit about how to publish models, what we would like you to do, how to get in touch with us. If you run into a problem, we're very open to anyone contacting us. So you can just email VCell support. We have a user group as well. So all of that information is here. Okay, so when you get to the main menu, we're gonna gonna work through the components, and first we're gonna talk about the physiology, how to build the model. So we start out with a single compartment, so we have to add what we need for our compartment. So I'm gonna click on this little donut tool that defines compartments, and what you'll see are these edges that are the ends of the compartment. If I click on one of them, I can either add a volume compartment or I can add a membrane. So let's add a membrane here. And let's add a membrane over here. And then we can change this. I have to change to the selector. I can change this to the plasma membrane. And I can double click and change this one to the site. So that's going to be the cytosol, and I can change this one to the nuclear membrane. And then I need another compartment to make the nucleus. So I'll add one here. I'll add a volume compartment. And I need an extracellular space, so I'll add a compartment out there. And again, I can change the names this time just to show you. I can change them down here in the properties pane. So I've selected this compartment. I see the properties related to that compartment. So this is going to be the extracellular space, and enter. It will change up there. And again, I can change the nucleus, name of the nucleus to bnuc. Okay, so now we have the compartments in space that our biomodel is going to incorporate: the extracellular, the plasma membrane, the cytosol, the nuclear membrane, and the nucleus. So now I need to add some species. So the species are the little green balls. So I can just take this and I'm going to add 2 species in this case. So let's make that RAN and RAN-C site. So let's label those. So one will be RAN. And one will
Excuse me,
be, yep.
ma'am, how can we define the compartment? What is compartment?
Compartment is a region of space that is generally in cells bounded by a membrane. Doesn't actually have to be bounded by a membrane, but it will have a surface. There'll be a flux within it. So you can think of it, the cytosol, because there's a membrane on one side, on between it and the extracellular space, and there's a membrane between the cytosol and the nuclear space, right?
All right, ma'am. It's mean we are considered cell organelles also as a compartment. We can take.
Yes, because there's a membrane. So molecules, for example, in the cytosol will not directly diffuse into the, it's not a common space. So it won't just directly diffuse into that. It has to cross the membrane, right? So whether it's the nucleus or the ER or any of the intracellular organelles, you would most likely consider them to be a separate compartment. Because you need a flux across that membrane. You need to define how it gets from one side to the other of the membrane. Does that make sense?
All right, ma'am. Thank you.
So, so this is going to be our cargo. And because, oh wait, I'm putting these in the wrong spot. Sorry, somebody should tell them. So I got to delete these species. So I select them and then just hit delete. Okay, because I was putting them in the extracellular space and they belong in the cytosol. So let's do that again. Okay. This is RAN. And this will be cargo. And I'm going to name this cargo underscore site. And the reason I'm doing that is because in virtual cell, if a species can only exist in in the compartment in which it's defined. Okay. And I can't have the same species in 2 different compartments. So cargo in the cytosol is literally a different species in the back end, in the mind of virtual cell is literally a different species than C in the nucleus. Okay. So I have to give them different names. Virtual cell will not allow you to name it the same thing. Okay. So I have my 2 species, RAN and C site, and they're going to bind together. So I need a reaction. So this is the reaction tool, this little line with an arrow. And so I can take RAN and make it a reactant. So it becomes, this little box becomes the reaction, and I can just draw the line again. And now it will actually make a product for me because I need a product as from this reaction. And then I can connect C site to this reaction. So now it becomes a binding reaction. So I have C site and I have RAN and I have the reaction. And S2, I'm going to rename as RAN-C, again in the cytosol. Okay. So, and then the reaction, I forgot what I used to call it, reaction. I want to be consistent here. So we name it. No, we just name it R. Okay. So I can just leave it as R0. It will list them R1, R2, R3, R4, or I can say, cargo binding, if that is easier for remembering. Sometimes when you get, a lot of reactions, having them all numbered is kind of annoying because you can't figure out what's where easily. Okay, so then I need this RAN-C site to flux through the nuclear membrane. So we have a tool for membrane flux. So that's here, and I'm going to put that flux in the membrane of the nuclear membrane, and then connect RAN-C. Oops, sorry. RAN-C is going to go to the flux, and then it's going to go into the nucleus. It will create a new species because there has to be some new species there. And S3 will be now RAN-C in the nucleus. Okay, so I've defined all the components that I need in this model. I've defined the different spaces, the different compartments, extracellular space, plasma membrane, cytosolic space, nuclear membrane, nucleus. And I've also described all the species, the RAN and the cargo, the complex in both the cytosol and the nucleus, and the reactions. So I have one binding reaction, okay, and I have one cargo. So again, here is where the arrows become important. So if you see these arrows in the reaction kinetics, these arrows define the forward rate, the positive flux of the reaction, okay? So the reverse rate would be the negative. So this doesn't mean that they're unidirectional, they're reversible reactions, however I define them, but it does mean that the positive flux is going to go in this direction. So actually, you'll see later that when this tutorial model was originally built, this reaction ran from RAN-C site to the component. So it will look a little different. So that just to remind me to point out that those arrows mean something when we're building these models in virtual cell. So if I select this reaction, again, the properties are down here in the property panes. And I can start out, in this case, it starts out with a mass action kinetics, and it gives me what would be mass action kinetics. So a forward rate constant times RAN times C site. So this is just exactly what Les explained in his reaction kinetics, minus the reverse reaction, right? So a constant for the reverse rate constant, and then RAN C, the concentration of RAN C, okay? So that's the overall flux, or J, defined as J, for this reaction.
Excuse me,
I
ma'am.
need, yes,
Sorry to interrupt, but I have
Yep.
2 questions.
Good.
Ma'am, in the toolbox, we see there are written like molecules and observables, but we can't see in the while creating the physiology. And the second thing, in when we defining the properties, there is a term called global, and ahead of expression there is written like units. How we input these units, and what are these global terms?
Okay, so I will explain global in just a minute. Global means, so if it's not checked, it means that this KF, for example, can only be used for this reaction. However, if it's global, if you say that you want that to be, I can't do it for mass action in this case. So if you want that particular rate to apply to multiple different reactions, to be able to reuse it in a different reaction, I have to define it as a different parameter. So I would change it here and say, make this maybe KRAN for RAN binding, okay? And then when K forward equals K RAN, I can then make it global. And so I could use it in some other reaction. If I had 2 different cargoes, I could use that same rate for binding, for example. Okay, and I can give it a value. So global parameters can apply to any of the reactions. Non-global parameters only apply to the reaction and can only apply to the reaction in which they're defined. Does that make sense?
Yep, ma'am.
Okay, so, and what was your other question? I'm sorry, I didn't quite get it.
The second question is, ma'am, there are written molecules and observables. What are these?
Yeah, molecules and observables, Michael will talk about when he does rule-based modeling. So there are specific terms that are used in rule-based modeling, where instead of defining a species, you define actually the molecules involved. So what we call a species here, let's say there's a phosphorylated form of RAN and a non-phosphorylated form. I don't really care, I just call it RAN. But you might care, and you might care exactly which site it's phosphorylated on. So in that case, you need to specify the actual description of the molecules involved. And that's that you can do in rule-based modeling. And Michael will talk about that. So I'm not going to talk about either molecules or observables today. Okay? But Michael will talk about it this afternoon.
Yeah, you can open, no, not Ann, but whoever is in audience, can open rule-based RAN tutorial in tutorials, and you will see that each of the species has molecules inside. Say RAN-C is a complex of RAN molecule and cargo molecule. Okay, and continue, please.
Right.
I'm sorry.
Okay, so
Yes, am I audible?
okay, so in this case, in order to stick
And there was another question, but I think the sound was very low.
okay, could you boost your sound a little bit maybe, or type it in the chat?
Yes, am I audible now?
Yeah, if you can speak even louder, it would be helpful though.
Oh, okay, just a minute, just a minute.
Yeah, that's better. Can you repeat your question?
Yes, so I had a doubt. I think I remember seeing the reaction going backwards from RAN side, C side to C side and RAN. So are we doing something here different, or does the direction change after we start the thing?
I'm sorry, your sound is still breaking up quite a bit.
It's very, very low. You probably need to adjust the gain of your microphone in the settings, in the audio settings.
Yeah, it's probably easier just to type question now.
Yes, is it okay now?
Yeah, that's better, much better. So, okay,
And continue talking. He's
okay.
like,
Yeah, you break in, Michael, because I sometimes get distracted. So once the question is there. Okay, so in this case, we do need to define what the forward and the reverse rate constants are, though. So in this case, I'm going to make the forward constant 1,000 per second. So the rate constants here in the units, that was another question that was asked. So because this is mass action kinetics, you know what the units must be in order to make that flux reaction balance, right? So in this case, it's going to be per second per micromolar for the forward rate constant. Okay. And the reverse rate constant, because it's a binding reaction, for the reverse rate constant, it's going to be, we'll put in a value here of 1. And this is going to be per second, right? So sort of independent of the concentration at this point, it's just per second, okay? And that depends on making this flux reaction balance, right? So the flux reaction is going to be micromolars per second. Now, if you need to define these separately, you can define them. You can change them. And the unit handling for the model itself is a little more esoteric. So you can change these if you need to, but I'm not going to talk about how to do that now. I can talk offline with anyone if they want to change the default units in VCell. So the default units are micromolar and second and micrometers. So that you can find if you go to the parameters, functions, and units, etc. There's this list on the biomodel menu. You can find all of these things. So these are all the parameters that we've described in the model so far. And the model unit system is listed here. So you can change that if you really want to, just be careful. To make sure that you're doing all the calculations properly. And most people will just leave it as is and do the conversion factors whenever they need to convert from whatever their parameters listed at to the model unit system that we're using. Okay. So let's go back to the reaction diagram. So now let's say I don't want this to be mass action. I want it to be something really complex. I can write any expression that I want. And so VCell has a number of different units that you can put in here. So I can do irreversible Michaelis-Menten reaction, which relies on knowing the Vmax for the equation and relies on knowing the VM, the Km, okay? So why did this give me an error? Okay, because if it's irreversible, so again, VCell tells you if there's something you're doing that's mathematically not correct. Okay, so irreversible Michaelis-Menten reactions must only have one reactant. So I'm not allowed to do that. Okay, so I can go back and say, okay, I actually want reversible. And so this would be the reversible rate constant. And that also, is that true? Okay. So, or I can just, so I can also just make this general, which means I can put in anything I want here. I can put in just 10, or I can put in some very complex reaction that's based on multiple things. If I want to, I can make it dependent on something else. So I can say that this rate, for example, let's put it some other species in here. So we'll say that this is some activator, some like IP3 or something that speeds up this reaction. If I want this to inform the reaction, I go back to the reaction. And in this case, I would here and say that it's some sort of forward rate constant times the new species I've got there. So let's say S4. So as you can see, there's a nice little drop-down menu that allows you to make sure you're picking what you think you're picking, times the concentration of RAN, times the concentration of C-site. Okay, so now this would be my flux reaction, okay? And KF, it's figured out, must be molecules per second micromolar to the -3 because I'm dependent on 3 different species, right? So, and you'll also notice that now there's a little dotted line drawn between the species and the reaction rate. Okay, so that's to remind you that even though it's not a participant, that S4 is acting like an enzyme, right? It's enhancing the rate of this reaction, but it's not participating in the reaction. It's not being changed by the reaction. So that makes this reaction also dependent on how much S4 there is in this molecule. Okay, but I don't want to do this. So I'm gonna try and delete this species, and you'll notice actually that I can't delete this species. It's going to say, no, you can't, because you've used that in somewhere in a reaction mechanism. So, and it tells you where in the cargo binding reaction. So I'm going to go back and go back to this reaction and get rid of that. Okay, so I'll get rid of the S4 term. And now I can delete this reaction. All right, so if things are used in the kinetics, it won't let you delete them until you change the kinetics. That's, again, just an important thing. And now if I go back, I want to go back to where I was, you'll find out that you can't. So you want to say, go back to mass action, but it's lost your rate constant. So VCell doesn't undo anything, right? If you change it, it's changed. And it doesn't save until you save it. So what you'll find is it's useful when you're sure you're at a spot that's correct to save the model. Okay, so I can save as, and I'll save it as delete. Done this before. And I'll save this as a model. And so now this part is all saved to the database. I can close it. I can go home. I can come back and just pick right up again. Okay. Okay. Thank you, Jim. I see there's a question. I'm just reading the questions. Yes, the reaction did go in the reverse direction, and I changed it from the tutorial. But again, just to point out that these arrows tell you the positive flux of the reaction are in the direction of the arrows. You can write it either way. And in the actual written tutorial, we've written it in the reverse way. Okay. So the question was also, does the direction change, which in the chat. So if you have defined the reaction to go in both directions, that is, there's a forward rate and a reverse rate, then it will go in both directions. And where the steady state will be will depend on the concentrations of the species. And until it reaches steady state, the fluxes will change, right? But once it reaches steady states, then the fluxes will be constant. Did I say that correctly, Pedro? Yeah. Okay, so then we also have a flux term here, right? And it's basically the same way. Oh, actually, I have to go back and redo my forward and reverse rates. So this is going to be 1,000. This is going to be 1. And actually, if you're following along in the tutorial model that is stored on, that you have access to, you'll see that I've just reversed these two numbers because I reversed the direction of the flux of this reaction. Okay, so then we have a flux reaction, and so we can again put in almost any terms that we want to in terms of how we define the kinetics for this reaction. So there's a lot of pre-built mechanisms here. The most common to use here is just a general flux density, and again we can put in then any expression that we want. And just want to make sure I do it the same way. So in this case, we're going to have a constant for the flux. So K flux times C site. What did I do wrong? K L times C site. And then minus ran C in the nucleus. So, okay, so there's a forward flux reaction. And as ran C builds up in the nucleus, it's going to slow down the flux. So the differential of the flux is going to determine the rate. So yeah, so then I have a rate constant here that I need to add in. I need to know what it is. And in this case,
Ann?
yeah.
Yeah, I think if you go back to that flux reaction,
What do I do wrong?
where's the rate expression? Yeah, it looks like you put the parentheses in the wrong place. You want to put it around the ran C minus ran.
Oh, yes, sorry.
Yeah. Just get rid of one of
No, it's automatically correcting that, Les.
well, go back in there and just
I want it here.
you want it there and not after site.
Sorry, yes, you're correct.
So now the forward and the reverse rates are the same and they're given by
Right.
KF.
Right. I thought that was okay.
Yeah, and one more.
Now I have an extra, sorry, parentheses are always my problem. Here we go. You have to be careful of parentheses when you're building complex expressions. Okay, so now it's correct, right? The flux times ran C site minus ran nuke. Okay, so that will define the flux across the nuclear pore. Okay, so now that's the full description of the biomodel, right? So you usually want to save things, okay? And because I have the full description of the kinetics and all of the species defined. But now, how do I want to solve this? What do I want to do with this mechanism, right? So now I can apply multiple different kinds of applications. So we can start with a new application, and we'll start with deterministic. And this is going to be application 0, and I can rename this as I choose. Just for convenience, I'm going to rename this as the PDE model because I'm going to start with a PDE, and I'll show you, start off as if I'm doing a PDE. So I happen to know that in this case, I do want a full model that includes diffusion. So I'm going to need a geometry. So the geometry I'm going to use for this model is going to be the same one that we use in the tutorial. So when you start out and create a deterministic application, it thinks it's going to be an ordinary differential equation. So it assumes, because it doesn't know any better, that you're just going to have different compartments and define a surface to volume ratio and size of each of those compartments, okay? So, you know, if you know how big the EC is and you know how big you want the nucleus to be and in volume, right, you could just add in those numbers here, okay? But I know that I want this model to represent a particular cell type, so I'm actually going to create the PDE geometry first. So I do that in geometry definition, and this afternoon I'll have a whole talk on how you deal with space in models. So I'm just going to very briefly provide you the means of doing this now. So I'm going to add a geometry, and I'm going to actually open it from one of the existing models. Again, this shows the utility of having a database here. Is I can go to this tutorial and I can use the already created tutorial for multi-app, which is right here. And I'm going to use that the geometry that was used in this tutorial, so I can just load it in. And this happens to be the geometry that we used in this tutorial. I can show a surface view like Les did, and it's a pretty complicated cell with a bunch of processes, and inside you can barely see the green in the nucleus, so I can make the nucleus little appear more. Okay, so it's got a nucleus. So I've in this is actually built from confocal microscope images. Again, it's a little complicated how we do this, so I'll show this in my spatial software this afternoon, spatial modeling component. But so the reason I'm going to put it in here is this cell, which is a real cell in the microscope, has, you know, a certain volume for the cytosol, a certain volume for the nucleus, a certain surface area for the membrane. And I want that to be perfectly represented when I do the ODE model to get to a steady state before I do my, my PDE calculation. So what I'm going to do is I'm going to include this model, and then now I'm going to take this PDE model and I'm going to copy it as non-spatial deterministic model. So it's going to copy everything that I have here as if it's an ODE. So I'm going to rename this and say ODE because I don't want to include space
excuse
here.
me.
Yep. Go ahead.
Yes, I have a doubt. I think I missed something in the middle. I'm not getting the applications part in my VCell. It's just showing 0 to me.
It's just showing you what?
I'm not sure if I missed anything. Yeah, right after the applications, it's showing 2 for you, but it's showing 0 for me. So I'm not getting any of the options like ODE, geometry, specifications, or anything.
So if you click on applications and you right click.
Yeah. Oh, okay.
So you'll see deterministic, stochastic, or network free, right?
Yes.
Right now, we're just doing deterministic.
Okay.
And the difference
got it.
between the PDE and the ODE is simply that the type of geometry that we're using. So when I added the geometry, this will automatically become because I've got a space defined that I want the simulation to be carried out in, it will be a PDE, okay? Because it has to account for the space. I copy that then as an ODE model, and in this case, I have no space. So the geometry definition is just one compartment. So I've assumed that everything is very well mixed. And then I can map all of these structures within that compartment. And did this work properly? Let me see. You will see that the sizes of each of these regions, let's see, is completely, oh, I haven't mapped them yet. That's why. Okay, never mind. Sorry, I did something wrong here. I'm just going to run through this again because I forgot to map it before I did it. So if I'm going to add the geometry again. So tutorials. Okay, so this now is going to be my PDE. So let's rename this as PDE. And what you have to do before you can do this in Virtual Cell is you have to actually map what pixels in this that you've selected belong to the cytosol and what belongs to the extracellular space, for example. So we have to do what's called structure mapping, where we take a line and go, so what I defined as EC in the biomodel is what's in red in the image, okay? So the image is segmented, and again, I'll explain all of the segmentation part this afternoon. And then the cytosol is what's brown, the nucleus is what's green, the plasma membrane is between the red and the brown, and the nuclear membrane is between the red and the green. And again, this also goes to let you know that the membranes in Virtual Cell are simply surfaces. We don't explicitly have a space for the membranes. They're simply the surface between each of the volume compartments. Okay, so that's why they're represented this way. So once I've mapped that, let's save that. You have to map compartments before you can save the model. And then if I copy this model as a non-spatial deterministic model, then this will become I can make this an ODE model, but what you'll see is it's taken exactly the right volumes for each of the compartments that are present in my geometry. So as Les said, it's usually much better to start out with an ODE based model, but just to get to steady state, to find your steady state, to see how the model's behaving, it's way faster. But ultimately, if you're pretty sure that you want to do a deterministic model, it can be very useful to start out from that geometry before you create your ODE model. Just because it means that it will calculate all of the sizes properly if you're doing it from an image. Okay, does everybody get that? So I have to move
Let me
along.
just add another comment to that.
Sure.
You may not realize it, but in that ODE model, the sizes matter. So even though it's not doing diffusion, the sizes of those various compartments and the surface areas of the membranes really do affect the concentrations, the things that you're after, the simulation results. I mean, an easy way to think about it is that for a given flux through the nuclear pores, if you have a very big nucleus, the concentration will be lower than if you have a very tiny nucleus, right? So the fluxes and the surface areas, all that stuff really matters. And that's why doing this using a real geometry to calculate the volumes and surface areas in an ODE model, a compartmental model, is really important.
So I'm going to rapidly run out of time here. So I just want to go through a little bit of creating an actual simulation from the ODE model. So the first thing that you need to do when you create a simulation, now I have the geometry. So in this case, it's a particular compartmental model with certain size ratios of all the compartments, right? So now I need to specify the components of this model, and in this case, the initial conditions. So I need to specify what the initial starting concentrations are. So in this case, I'm going to just specify, I'm going to start with everything as the complex of RANC and the nucleus. Let me just get my cheat sheet up here. And so I'm going to start with everything as RANC in the nucleus, and I'm going to make this be e to the -4. And the reason I've done it this way is that we just want to make the tutorial model consistent with the make small numbers of molecules so that when we do the spatial stochastic, it doesn't take too long. So that's a pretty small number of molecules. But I'm going to start with everything in the cytosol, and that's really all I need to do in order to now go and create a simulation. So the simulation, I just add a simulation, and let's say we're going to run this for, I need to edit the specifics of the simulation. So let's go to the simulation, and I can, these are the parameters. I can change them if I want to, but mostly I want to look at the solvers. So these are a bunch of different solvers that we have. Usually VirtualCell will pick the best one for your model situation. It's, if you're not sure, just stick with what VCell chooses. And we do have a little description of each of the solvers so you get an idea. These are usually built by other people, so you can look up in the literature exactly how they work. But we have a short description for each one. And then you need a start time and an end time. So this one, we're going to go for 10 seconds. And the time step, you don't need to choose. Usually the solver will choose the appropriate time step for how fast the reactions are occurring. So it's appropriate to your situation, how often it will do the calculations. But what you do want to think about is how often you want to keep the results. So you don't have to keep all of the results that were solved in the solver. And especially if you want to align it with some data that you have, you usually want to put something in, not save every single step, but you might want to save like every second or something like that. So if you change this to 1, it will save every second. Let's do it to every 0.2 to be consistent. Yeah. Okay. So that means it will make sure that it solves at exactly 0.2 seconds as well. And it will also only save the data when it solves at every 0.2 second of the solution. Okay. And so then this blue button will run a native run on your own computer. So you can select that. And it'll start the solver on your own computer, and it will solve it pretty quickly. And you will get solutions. These are not saved anywhere. So as soon as you close this window, they're gone forever. You'd have to open them up, solve it again. Okay. So we usually use these for quick runs when you don't want things to go back and forth through the database. But in general, you would click this green button, and that will send the problem to the to our solvers here at UConn. The other thing to point out that you can see the math. Once you specify the application, you generate math. And if you look at the math description language, this is actually the math that's going to be solved by that solver. Okay, so you can create a math model from that, or you can by editing the math description language itself. And then the rest of this, I'm going to turn over to Pedro now, and I'll talk a bit about all of the tools that you can use to analyze simulations later when I talk about spatial simulations. Okay, any further questions? I do have to stop now because it's Pedro's turn.
Thank you, Anne. I'm
Okay.
gonna
Grab the screen.